Hello everyone, thanks for being here today listening to this presentation. My name is Renan Favre, and today we're going to talk about the Auxin herbicides and the current status and mechanism and mode of action. This presentation is based on Klaus Grossman review article. So, phytohormones overview. So we know that plant hormones, they control the development, stress responses, and growth. And in the main groups, we have auxins, gibberellins, cytokinins, ethylene, and abscisic acid. And the auxins, they interact with other hormones in complex networks. And that's why auxins, they are known as the master hormone. The indolacetic acid is the principal natural auxin in higher plants and has influence on plant growth and development. And also, the auxins, they direct the tropisms and initiate vascular differentiation, which is xylem and phloem. And here are some functions of auxins. We have the regulation of cell division and elongation, vascular tissue differentiation, floral meristem differentiation, leaf initiation, and apical dominance and root formation. And also, auxins, they play a role in senescence and tropic responses. In this picture, we can see the light. In the left side, we can see the light coming from the top, and then the auxin spreads equally down both sides of the plant. And in the right side, we have the light coming from the right side, and then the auxin collects on the shady side. And how the auxins they came from growth hormones to herbicides. So basically, the synthetic auxins, they were developed in the 40s. For example, we have 2,4-D and MCPA, and they mimic indolacetic acid, but they are more stable and longer acting inside the plant. Okay, and they are used as selective herbicides in cereal crops, and that's what happened in the 40s. The scientists, they were trying to develop something to increase the yield of cereal crops, and then they got 2,4-D, which was amazing at that time. However, later, well, there's a little bit of history here, okay? So, later, at the Vietnam War, they started using like 2,4-D and the other herbicides to defoliate the battlefield to have a better view, right? So, yeah, a little bit of history here. And as I was saying, they are used as selective herbicides in cereal crops. However, we also have resistance for some of our broadleaves as well. For example, in list E3 soybeans, which is resistant to 2,4-D, glyphosate, and glufosinate, and also X10 flax soybeans, which is resistant to dicamba, glyphosate, and glufosinate. This is the chemistry of auxin herbicides. We have these 5 main families, which is phenoxycarboxylic acids, that's 2,4-D and MCPA, benzoic acids, dicamba, pyridinic carboxylic acids, bichloran and clopyrrolate, quinoline carboxylic acids, where we have quinclorac, and aromatic carboxymethyl derivates, macroprop. And also we have these erypiconolates, which is another family that arrived in the market in 2017. As a common name, we have fluoroperoxyfenbenzyl. Okay, and here are some of the structures of the herbicides. For example, in the left side, we have MCPA, dicamba, bichloran, fluroxypyr, dichloroprop. This is something that we don't need to memorize, right? But sometimes it's good to have a better view of how these structures are for a better understanding, right? All right. So when applied as herbicides, the high auxin levels, they overstimulate the growth pathways inside the plant, which results in disrupted growth, deformities, and tissue decay. and has similar. It is similar to effects seen in dolosidic acid overproducing transgenic plants. And for example, we have uh, for soybeans, one of the benefits we have the improved nodulation and early seedling development, right? So that's a good benefit about transgenic plant which overproduces dolosidic acid. And this is a phrase. This is a good phrase from Gilbert F.A. saying that Auxin herbicides cause susceptible plants to grow themselves to death. Uh, so in this whole process, we have three phases. Phase one is the stimulation, which occurs within 8 hours. So in the first 2 hours, we have the activation of ethylene biosynthesis via ACC synthase, right? And then after 3 to 4 hours, we have the deregulated growth, causing leaf apnasty, tissue swelling, and initiation of stem curling. Oh yeah, just for your information, this ACC synthase is not the same as the ACCase, okay? Because this ACC synthase is responsible for ethylene biosynthesis. It is related to ethylene biosynthesis. And then after 5 to 8 hours, we have the abscisic acid accumulation inside the plant. All right, then we go to phase number 2, which is called inhibition within 8 to 24 hours, where we have the shoot and root growth slows and leaf pigmentation increases. We have stomatal closure, lower transpiration and photosynthesis, and reactive oxygen species overproduction, which leads to stress and damage. All right, and finally, at phase 3, we have the senescence after 24 hours. So we have an accelerated foliar senescence with chloroplast damage and progressive chlorosis, and also the destruction of membrane and vascular system integrity, leading to tissue desiccation, necrosis, and eventual plant death. All right, and here are some of the phytotoxic effects of the Auxin herbicides. So the herbicidal auxins, they mimic the natural plant hormone but at a supraoptimal concentration, which leads to overstimulation of growth processes and disrupts the normal metabolic and growth functions, causing plant death, right? And here are the molecular targets. We have the auxin receptors, which are responsible for gene expression change, and also the membrane ion channels, which alters the ion balance, affecting cellular processes, and also the plasma H plus minus ATPases, causing cell elongation, uncontrolled growth, and tissue damage. And here we're gonna get deeper on molecular biology now. All right, we're going to talk about auxin perception, signaling, and gene expression. So for the auxin receptors, the mechanism of actions, we have that early research identified auxin binding protein 1, which we are going to call ABP1, as a candidate receptor due to its auxin binding activity. All right, it is responsible for fast auxin responses like cell expansion, and also TIR1, which stands for Transport Inhibition Response 1. It was later found to be the receptor that helps plants detect auxin, and it is part of a protein called AC. Sorry, it is part of a protein group called SCF, which helps break down the other proteins in other cells. So basically, the auxin binds to TIR1, stabilizing its interaction with auxin and dolosidic acid repressor proteins, which leads to the degradation of auxin and dolosidic acid proteins by the 26S proteasome. All right. So here we have this picture about the phases again. So after 5 hours, we have the stimulation phase, where we can observe stem curling, tissue swelling, leaf epinesty. And then after 24 hours, which is the inhibition phase, we have an intensified green leaf pigmentation, stomatal closure, reduced transpiration, and carbon fixation. Finally, after 72 hours, we have the decay phase, senescence, and cell and plant death, right? Where you have the disruption of chloroplasts, membranes, and vascular systems. Here are the interactions between the hormones and also the growth response. Okay, so for TIR1 and ABP1 receptors, these receptors they bind the auxin herbicides or indolacetic acid at a supraoptimal concentrations, which leads to overexpression of auxin responsive genes. And this whole process triggers to a cascade of biochemical and physiological events. All right, and also we have the ethylene formation when the auxin-induced ACC synthase activity results in elevated ethylene production. The ethylene production leads to lateral cell expansion, leaf abscission, and also epinesty. Right, and then we have the abscisic acid biosynthesis because the auxin herbicides they stimulate the abscisic acid accumulation by activating NCED genes, and these NCED genes they are responsible for abscisic acid biosynthesis, okay, which leads to growth inhibition, stomatal closure, and oxidative stress. And this is what happens when we have the phytotoxic syndrome, because the combined effects of auxin activity, ethylene, and abscisic acid result in the phytotoxic syndrome, right? And these mechanisms they explain the growth inhibition, senescence, and tissue damage observed in susceptible plants. And over here we have this picture about how everything works. So basically, when the auxin is low inside the plant, the auxin and indolacetic acid protein repressor proteins they bind to ARF, and then when the auxin is in high levels inside the plant, they don't bind because those proteins they are degraded by the 26S protease, right? So once degraded, we have the ARF free to produce gene activation. And when ARF produces gene activation, it increases the levels of NCED and also ACS. So when the NCED is in high levels, it leads to increase in the levels of abscisic acid, which leads to senescence, tissue decay, growth inhibition, and all that. All right. On the other side, when the ACS is high, it leads to increase of ethylene levels inside the plant as well, which also leads to leaf epinesty, tissue swelling, senescence, inhibition of and also the inhibition of auxin transport. All right. And for some sensitive grasses, we have this overproduction of hydrocyanide, right, which leads to cell death and tissue necrosis. However, most of the grasses or tolerant crops they have high levels of beta CAS, which is a protein responsible for hydrocyanide degradation, right? So why don't auxin mimic herbicides kill monocots? Well, we have some answers for this. So first one, receptor sensitivity. In dicots, we have TIR1 and AFB receptors highly responsive to auxin herbicides, which leads to growth deregulation. However, in monocots, they are less sensitive and different receptors, which leads to minimal gene activation. And also, um, when using auxin herbicides in the cuts, we have increased levels of ethylene biosynthesis and ROS, which leads to senescence and death. However, in monocots, they don't trigger this cascade, which avoids damage. And also, some monocots, they are efficient in detoxification because they produce more beta CAS, as I said before, which is a protein responsible for cyanide detoxification, which protects from cell death, right? However, there is an exception among the auxin herbicides called quinclorac, which also control grass weeds, right, like Echinocloa, Digitaria, Setaria, and Brachiaria. And there's a key mechanism for this, which is the cyanide toxicity. So basically, quinclorac stimulates ethylene biosynthesis, which leads to production of hydrogen cyanide as a co-product. And then the ACC, which is an ethylene precursor protein, is made in roots and transported to shoots. In the shoots, cyanide accumulates, leading to leaf chlorosis, wilting, and shoot necrosis. And these are the phytotoxic effects of cyanide. Because cyanide, or hydrocyanide, it inhibits key enzymes like cytochrome C oxidase, nitrate reductase, ribisco, and superoxide dismutase, resulting in respiration inhibition, photosynthesis disruption, and reactive oxygen species buildup, and also the cell and plant death. However, as I said, for tolerant species, for example, rice, they have a low ACC cyanide accumulation and high levels of beta CAS, which is a detox enzyme. And here I have some examples. For example, this is one of my fields where we sprayed this herbicide called Nova Grace, which has in its composition 2,4-D and fluoroperoxifene benzyl, both group 4 herbicides. So as we can see a lot of apenacity here. We can see a lot of speckling, tissue swelling, and all that. This is the most characteristic symptoms that we're going to see of auxin herbicides, right? We have a lot of stem elongation here. We have a lot of cell division and a lot of apenacity, right? Yep, that was it. Thanks for listening to this presentation. Have a good day.